Okay, so let's start to talk about the TCA cycle. The TCA cycle is a cycle in the mitochondria that's going to generate a lot of ATP for your cells. And so carbons have to enter the TCA cycle, and one of the major sources of carbons is going to be activated acetate, or what we also call acetyl-CoA. So acetyl-CoA is going to be made by our fuel sources, such as our fatty acids, when you break down fatty acids, ketone bodies, acetoacetate in particular, and then as our sugars go through glycolysis, they are going to converge on pyruvate. Also, amino acid alanine can be converted to pyruvate, and of course, our alcohols can also generate acetyl-CoA and enter the TCA cycle. So the movement of the carbons in acetate is through the TCA cycle is not necessarily going to end in making energy. There are other reactions that can take place, and so these are called anaplerotic reactions. And so just keep in mind that if the cell does not need energy, it has to do something else with the carbons that it has. And so it can start building things. It can make fatty acids. It can make amino acids. In certain areas, such as the brain, our components of our TCA cycle can actually form neurotransmitters. If you're going to be making heme, succinyl-CoA is going to be a precursor for that, and that is part of the TCA cycle. And also other amino acids can come from oxaloacetate as well as making glucose, so gluconeogenesis. So again, it's just going to depend on the needs of the cell at the time as to where the carbons entering the TCA cycle will eventually end up. Now, pyruvate is going to be a major source of activated acetate because pyruvate is going to be formed from glycolysis. And pyruvate is actually going to be the form that the carbons come into the mitochondria. So this can enter the mitochondria. Now, pyruvate is a 3-carbon compound, and acetyl-CoA is going to be a 2-carbon compound. And so pyruvate basically is going to either generate a 2-carbon compound or a 4-carbon compound. And this, again, depends on the energy needs of the cell. If the cell needs energy, you're going to make acetyl-CoA. And this is a 2-carbon compound. If the cell does not need energy, your pyruvate is now a 3-carbon compound is going to become a 4-carbon compound of oxaloacetate. And so this is going to require the cofactor of biotin. And biotin is going to be linked to your compound through a lysine residue. Now, basically what biotin is going to do, it's going to grab your ATP and it's going to grab your carbon dioxide, which is typically going to be a bicarbonate. And it's going to transfer this carbon in the bicarbonate to your 3-carbon pyruvate to generate the 4-carbon oxaloacetate. Now up here, if you're going to become acetyl-CoA, you're going to go through the pyruvate dehydrogenase. And the dehydrogenase is going to do the opposite. It's going to release a carbon, and it's going to generate a 2-carbon. So pyruvate is a 3-carbon, which is a major source of carbons coming into the TCA cycle or into the mitochondria via glycolysis, is going to have to become either a 2-carbon acetyl-CoA and be pushed into the TCA cycle, or become a 4-carbon oxaloacetate. And as oxaloacetate, it can do many things, including go into the TCA cycle if now the cell needs energy. Now, there's also other ways you can bring in carbons into the TCA cycle. A very quick way is that you can generate pyruvate from alanine. So some amino acids can directly make pyruvate, and then pyruvate will go into the same direction that we just discussed, where you can add a carbon to it, and this will generate oxaloacetate. Now, aspartate can also become oxaloacetate, and this is going to be through a transamination reaction. So this is a TA reaction. You can have other amino acids become fumarate. You can have amino acids break down to glutamate, and this can become alpha-ketoglutarate and enter the TCA cycle. And then you can have your odd-chain fatty acids generate propionyl-CoA, which will eventually become succinyl-CoA, and your branch-chain amino acids, valine, isoleucine, can also become propionyl-CoA, and then this can become succinyl-CoA. So there are many ways, again, that your carbons can enter the TCA cycle if your body or your cell needs energy. So with respect to the TCA cycle, there's going to be a lot of negative delta Gs, and so this is going to really push the cycle forward. So we have some critical points in the cycle. We have our oxaloacetate, which is our 4-carbon, and this is going to merge with our activated acetate, our 2-carbon, and this is going to form our 6-carbon citrate. And so this step is a rate-limiting step. And then you're also going to have, and this is going to be catalyzed by citrate synthase. You're also going to have isocitrate become alpha-ketoglutarate. And this is going to be isocitrate dehydrogenase that does this. And we're losing a carbon here. So here we're going from a 6-carbon. Now we're going to a 5-carbon. And then the next step is also going to be regulated, and it's going to be important. It's alpha-ketoglutarate to succinyl-CoA. We're losing another carbon here. And so this is going to be a 4-carbon. Now, as we go through the TCA cycle, we're going to talk about how each of these steps are going to be regulated. But at this point, you just need to understand that we started with a 4-carbon oxaloacetate, and by succinyl-CoA, we have regenerated that 4-carbon. And the rest of the steps through the TCA cycle are going to be moving around carbons and also generating ATP, of course. Now, there are some important coenzymes of the TCA cycle. So we're going to have NAD and FAD that are going to be able to pick up electrons, and different enzymes use these. We also have citrate synthase that uses the CoA derivative of acetyl-CoA. We also have some complexes, our alpha-ketodehydrogenase complex. This is going to use many different coenzymes: thiamine pyrophosphate, lipoate, FAD. And then it's also going to have non-bound coenzymes of NAD and CoA with a thiol group. And so this one compound uses a lot of those, which means that each of these are going to come, of course, from vitamins. Now, finally, we just have to talk very briefly about some enzyme deficiencies. These are going to be deficiencies in enzymes that are linked to mitochondria DNA. And so we have our isocitrate dehydrogenase genes 1 and 2, and this has been linked to gliomas in brain. We also have our succinate dehydrogenase genes A and B through H, but C, D, and H are going to be important. And then there's also a succinate dehydrogenase assembly factor, which is the AF2, and these have been associated also with cancers. And then fumarase gene, a fumarase hydratase gene, has been associated with leiomyomas and type 2 papillary renal cancers.